All right, so today we're going to try to tackle something pretty complex.
Yeah.
Nicotinic acetylcholine receptors.
Exceeders.
NCRs. You've sent over a ton of material on this, and
A lot.
I have to admit, even I was a little intimidated when I saw it, but I think we can break it all down
Absolutely.
into something understandable.
We can think of this as like a deep dive, express lane to understanding these fascinating receptors.
Okay, perfect.
How they work, why they're important, even how they're targeted by medications.
So first things first,
Yeah.
can you give us a quick overview of what exactly nAChRs are?
They're receptors found all over the nervous system,
Okay.
and you can imagine them kind of like tiny little gateways on nerve cells.
Okay.
And their job is to respond to a very specific signal, the neurotransmitter, acetylcholine or acetylcholine.
Acetylcholine. Okay, I've heard of that.
Yeah.
Yeah, that's like a chemical messenger between nerves, right?
Exactly. When AC8 binds to an nAChR, it's like a key fitting into a lock. This binding triggers the receptor to change shape, which opens up a channel through the cell membrane.
And what happens when that channel opens?
That's when things get really interesting. Charged particles, mostly sodium ions,
Okay.
or Na+, and sometimes calcium ions, K2+, can rush into the cell.
Okay.
This influx of ions changes the electrical charge inside the cell, and that's what kicks off this whole cascade of events.
So it's like a domino effect.
Yeah.
The ACH binds the channel, opens, ions flow in, and boom, a signal is sent.
Precisely. Now, here's where it gets a little more complex. nAChRs aren't all identical. They're actually made up of 5 protein subunits, and these subunits come in different flavors,
Right.
often a mix of alpha and beta types.
So it's not just one type of ACR doing everything.
Not at all. The specific combination of subunits in a receptor determines where it's located in the body and how it functions.
So like a modular system.
It's like a modular system that creates specialized receptors for different tasks.
Okay, I'm starting to see how intricate these receptors are.
Yeah.
So let's get specific. Where in the body do AChRs have the biggest impact?
Well, one of the most critical places is the neuromuscular junction, the meeting point between a nerve and a muscle.
So that's how our brains tell our muscles to move.
Exactly. When a nerve impulse reaches the end of a motor neuron, it releases acetylcholine into that junction. The ACR then binds to ACIs on the muscle fiber, and that triggers a rapid influx of sodium ions.
And that's what causes the muscle to contract.
Yes.
So every time I move, I have these tiny receptors to thank.
You got it.
Wow.
But their role doesn't stop there. ACIRs are also incredibly important in the central nervous system, which includes your brain and spinal cord.
Okay, and here's where it gets really, really interesting.
Right.
What are they doing up there?
They're involved in all sorts of cognitive processes, from paying attention and learning to forming memories and even regulating mood.
Wow, so these little receptors are involved in my thoughts, emotions, and memories.
Yeah.
That's incredible.
It is, isn't it? And their activity in the brain helps explain why nicotine, which also binds to nAChRs, can have such powerful effects on cognition and, unfortunately, addiction.
Oh, so nicotine is basically hijacking those receptors, pretending to be acetylcholine.
Exactly. And because NACA are so crucial for so many essential processes, they've become targets for a whole range of medications.
That makes sense. So how are nAChRs being used in the development of drugs?
Well, for example, some drugs that block nAChRs at the neuromuscular junction are used as muscle relaxants during surgery.
So they're temporarily paralyzing the muscles by preventing acetylcholine from binding.
Precisely. But then there are other drugs that are being researched that actually enhance nAChR activity in the brain.
Oh, interesting.
These are being explored as potential treatments for Alzheimer's disease and other cognitive disorders.
So boosting the activity of these receptors could potentially help improve memory and cognitive function in people with these conditions.
That's the hope.
That's amazing.
It's a complex area of research, but it shows just how much potential there is in targeting nAChRs for therapeutic purposes.
Okay, so we've talked about their structure, how they work, where they act in the body, but we haven't really touched on the specifics of those subunits you mentioned,
Right.
the alpha and beta types.
Yeah.
What can you tell us about those?
Well, that's where things get really interesting. It turns out that the specific combination of alpha and beta subunits in an AChR can drastically affect its properties,
Okay,
like how sensitive it is to acetylcholine or how long the channel stays open.
so it's not just about whether it's alpha or beta,
Right.
but about the specific arrangement of these subunits.
Exactly. For example, some nAChRs are made up entirely of alpha subunits,
Okay,
while others have a mixture of both alpha and beta. And within each type, alpha and beta, there are even further subtypes, each with its own unique properties.
wow, that's a lot of variation.
It is.
What does all this diversity mean in terms of how these receptors function?
Well, for one thing, it means that different types of nAChRs can respond differently to the same signal.
Okay.
Some might be very sensitive to acetylcholine, triggering a response with just a small amount of the neurotransmitter.
Okay.
Others might require a much higher concentration of nAChR to activate.
So it's like having different volume controls on different receptors,
Yeah.
allowing for fine-tuning of the signals.
That's a great way to think about it. And this fine-tuning is essential for the nervous system to function properly.
Oh, okay.
It allows different parts of the brain and body to respond to acetylcholine in a way that's tailored to their specific needs.
So this subunit diversity basically expands the repertoire of nAChRs,
Yeah,
making them even more versatile and adaptable.
exactly. And this adaptability is what makes them such fascinating targets for drug development. By targeting specific nAChR subtypes,
Okay.
researchers hope to develop more precise and effective treatments for a wide range of conditions.
That makes a lot of sense. So not only are nAChRs incredibly important for how our bodies function, but understanding their nuances could be key to unlocking new therapeutic avenues.
Absolutely. And that's why research into these receptors continues to be such a vibrant and exciting field.
This is all incredibly fascinating. But I'm curious,
Yeah.
when it comes to those ions flowing through the channel,
Mm-hmm.
is it always just sodium? Or are there cases where calcium plays a more prominent role?
That's a great question, and it highlights another layer of complexity in how nAChRs function. While sodium is the primary ion involved in most nAChR sodium-mediated signaling,
Okay.
there are certain subtypes of these receptors that are more permeable to calcium.
So the specific combination of subunits can also influence which ions are allowed to pass through.
Precisely. And this distinction between sodium and calcium permeability is particularly important in the central nervous system,
Okay.
where calcium plays a crucial role in various cellular processes,
Okay.
including neurotransmitter release and synaptic plasticity.
Ah, synaptic plasticity.
Yes.
That's the ability of synapses,
Yes.
the connections between neurons to strengthen or weaken over time.
Exactly.
Isn't that essential for learning and memory?
Exactly. And the fact that some nAChRs can allow calcium into neurons means that they can directly influence synaptic plasticity,
Wow.
contributing to the brain's ability to adapt and change in response to experience.
So these receptors aren't just relaying signals.
Right.
They're actively shaping the very connections that make up our neural networks.
It's really quite remarkable, and it underscores just how important these seemingly simple receptors are for the complexity of brain function.
This is all incredibly insightful.
Good.
But before we move on, I want to make sure I have this straight.
Okay.
We've talked about how nAChRs are essential for muscle contraction
Mm-hmm.
and for a whole range of cognitive processes.
Yes.
But can you give us a concrete example of how these receptors are actually working in our bodies on a daily basis?
Absolutely. Let's take the simple act of reaching for a cup of coffee. That seemingly effortless movement actually involves a complex interplay of signals between your brain, your nerves, and your muscles, and nAChRs are right at the heart of it.
Okay, walk me through it. What's happening at the level of these receptors?
Well, first, your brain sends a signal down your spinal cord to the motor neurons that control the muscles in your arm and hand.
So the brain is initiating the movement command?
Yes.
Then what?
When that signal reaches the neuromuscular junction, remember that's the meeting point between the nerve and the muscle.
Right.
The motor neuron releases acetylcholine into the synapse.
Okay, so the chemical messenger is released.
Yes.
And that's where the nAChRs come in.
Exactly. The acetylcholine molecules then bind to nAChRs on the surface of the muscle fiber, causing those receptors to open their ion channels.
And that allows sodium ions to flood into the muscle cell, right?
Precisely. That influx of sodium ions changes the electrical charge inside the muscle cell, which triggers a cascade of events that ultimately leads to the contraction of the muscle fibers.
And that contraction is what allows you to lift your arm and bring that cup of coffee to your mouth.
Exactly. So in that simple act of reaching for your coffee, you've engaged countless nAChRs, each one playing a precise role in coordinating the movement.
Wow. I never realized how much was going on at the molecular level just to perform a simple action.
Yeah.
It's amazing to think that these tiny receptors are working so hard behind the scenes to make our everyday lives possible.
It really is. And it highlights the incredible power and precision of the nervous system.
Okay, so we've covered a lot of ground on nAChRs.
We have.
But I'm curious, are there any other interesting facts or insights about these receptors that we haven't touched on yet?
Well, one thing that I find fascinating is that nAChRs aren't just found in humans. They're actually present in a wide range of organisms,
Oh.
from simple invertebrates to complex mammals.
So they're a pretty ancient and evolutionarily conserved type of receptor?
They are indeed. And this suggests that they play a fundamental role in nervous system function across a wide variety of species.
Makes sense.
Yeah.
If something works well,
Right.
evolution tends to hold on to it.
Exactly.
Are there any other notable differences between HERs in different species?
Well, one interesting example is that some venomous creatures like snakes and spiders have evolved toxins that specifically target HERs.
Oh, wow. So they use these toxins to paralyze their prey.
Exactly. These toxins can either block HERs, preventing them from responding to acetylcholine,
Okay.
or they can overstimulate them, leading to muscle fatigue and paralysis.
That's both fascinating and a little terrifying.
It is.
It's incredible how evolution has found ways to exploit these receptors for such diverse purposes.
It really is. And it highlights the importance of understanding the intricate workings of nAChRs, not just for human health, but for understanding the diversity of life on Earth.
I think we've covered a lot of ground in this first part of our deep dive into nAChRs. From their structure and function to their role in muscle contraction, cognition, and even venom, these receptors are clearly essential players in the drama of life.
I agree. And as we move forward in our exploration, we'll delve even deeper into the world of nAChRs, uncovering their connections to other biological systems and exploring the potential they hold for future medical breakthroughs.
I'm excited to continue this journey with you. But for now, let's take a quick pause and give our listener a moment to digest all this incredible information. We'll be back soon with more fascinating insights into the world of nicotinic acetylcholine receptors.
Welcome back.
So we've learned so much about nAChRs already.
Yeah,
It's amazing how much more there is to uncover.
exactly. And one of the things that makes them even more fascinating is how they're connected to other biological systems.
Ah, so they don't just operate in isolation.
Yeah.
They're part of a larger network.
Precisely. One example of this interconnectedness is the relationship between nAChRs and the POMC system.
Okay, POMC.
Yeah.
That rings a bell from some of the material, but refresh my memory. What exactly is POMC?
POMC stands for propionmelanocortin.
Okay.
It's a large precursor molecule that gets chopped up into smaller pieces, and those pieces become a variety of hormones and neuropeptides.
Hormones and neuropeptides, those are like chemical messengers that travel throughout the body, right?
Exactly. And some of the key players that come from POMC actually interact with the systems that nAChRs are involved in.
So this POMC system can indirectly influence how nAChRs function.
That's right. It's not a direct interaction, but more of a modulation. One of the most interesting examples of this is the role of melanocortins.
Melanocortin, those are involved in pigmentation, right? Like
Yeah.
determining skin and hair color.
Yes, but they have a much wider range of functions than that. They're also involved in things like appetite regulation, energy balance, and even stress response.
Wow, it's quite a diverse portfolio.
Yeah.
But how do they tie back to nAChRs?
Well, melanocortins have their own set of receptors called melanocortin receptors, or MCRs.
Okay.
And the activity of these melanocortins through their receptors can actually influence the release and signaling of acetylcholine.
Ah, I see. So the melanocortin system can essentially tweak the activity of the cholinergic system,
Right.
which is the system that uses acetylcholine as its messenger.
Precisely. And this interplay between these two systems highlights the complex web of interactions that's constantly at play in our bodies.
It's like a delicate dance between different signaling pathways,
Yeah,
each one influencing the others.
that's a great analogy. And it's this intricate choreography that allows our bodies to maintain balance and adapt to changing conditions.
This connection between ATRs and the POMC system is fascinating, but I'm wondering,
Yeah.
what are the practical implications of this relationship? Does it have any relevance for things like drug development or understanding certain diseases?
Absolutely. For example, researchers are investigating whether targeting the melanocortin system could be a way to indirectly influence HER activity.
So instead of directly targeting the HERs themselves,
Right.
you could potentially modulate their activity by manipulating the melanocortin system.
Exactly. And this could be particularly useful in cases where directly targeting HERs might have unwanted side effects.
That makes a lot of sense. It's like finding a backdoor to influence a system without having to go through the main entrance.
That's a great way to put it. And this approach is being explored for conditions like addiction, pain management, and even obesity.
Wow. So this seemingly obscure connection between nAChRs and the POMC system could have far-reaching implications for treating a wide range of conditions.
It's certainly a promising area of research, and it underscores the importance of understanding these complex biological interactions as they often hold the keys to unlocking new therapeutic avenues.
Speaking of therapeutic avenues, one of the things that really stood out to me in the research I sent over was the potential of nAChR targeting drugs for treating Alzheimer's disease.
Yeah.
Can we delve a little deeper into that?
Absolutely. Alzheimer's disease is a devastating neurodegenerative disorder that affects millions of people worldwide.
Right.
And one of the hallmarks of this disease is the loss of cholinergic neurons,
Okay.
which are the neurons that use acetylcholine as their neurotransmitter.
So in Alzheimer's, there's a depletion of the very messenger that nAChRs are designed to respond to.
Exactly. And this loss of cholinergic signaling contributes to many of the cognitive symptoms of the disease,
Okay.
such as memory loss, confusion, and difficulty with learning and attention.
That makes sense.
Hmm.
So how are nAChR targeting drugs being used to address this problem?
Well, the idea is that by enhancing the activity of the remaining nAChRs, you can potentially compensate for the loss of acetylcholine and improve cognitive function in these patients.
So it's like trying to boost the signal strength even though the transmitter is weaker.
That's a good analogy. And there are a few different ways that researchers are trying to achieve this. One approach is to develop drugs that directly activate nAChRs, mimicking the effects of acetylcholine.
So these drugs would essentially act as substitutes for the missing acetylcholine.
Exactly. Another approach is to develop drugs that prevent the breakdown of acetylcholine in the brain, thereby increasing the amount of this neurotransmitter that's available to activate nAChRs.
So instead of replacing the missing acetylcholine, you're trying to make the existing supply last longer.
Precisely. And there's also research being done on drugs that can increase the production of acetylcholine in the brain, essentially trying to replenish the depleted supply.
Wow. So there are multiple strategies being explored to target the cholinergic system
Yeah.
and enhance nAChR activity in Alzheimer's patients?
There are. And while there's still a lot of research to be done, there's hope that these anti-R targeting therapies could offer new ways to slow the progression of Alzheimer's disease and improve the quality of life for those affected by this devastating condition.
It's encouraging to hear that there's so much effort being put into finding new treatments for Alzheimer's.
Yeah.
It's a disease that touches so many lives, and any progress in this area is incredibly valuable.
I agree. And the fact that nAChRs are involved in so many different aspects of brain function makes them a promising target for a wide range of neurological and psychiatric disorders, not just Alzheimer's.
So understanding these receptors could be key to unlocking new treatments for a whole host of conditions.
It's certainly a possibility. And that's why research into nAChRs continues to be such a vibrant and exciting field.
It's clear that we've only just scratched the surface of this complex and fascinating world. I'm eager to see what else we uncover in the final part of our deep dive.
Me too. There's still so much to explore, from the intricate details of nAChR structure to the potential these receptors hold for future medical breakthroughs.
I'm ready for the next stage of our exploration. But for now, let's give our listener a moment to reflect on all that we've learned so far. Okay, so we've talked about the structure of nAChRs,
Yeah.
how they work, their role in the body, even their connection to other systems like POMC.
Right.
But I'm curious, how did scientists figure all this stuff out in the first place?
That's a great question. Unraveling the mysteries of nAChRs has been a journey of scientific discovery spanning decades.
Wow.
It's a story of meticulous research, unexpected twists and breakthroughs that have really revolutionized our understanding of the nervous system.
Okay, I'm ready for a story time.
All right, well, I think a good place to start is with the discovery of acetylcholine itself.
Okay.
Back in the early 20th century,
Okay.
scientists were just beginning to understand that chemical messengers, what we now know as neurotransmitters,
Okay.
played a role in communication between nerve cells.
So before that, they didn't even know how nerves were talking to each other.
Not exactly. There were theories, but the idea of chemical transmission was a major breakthrough, and one of the pioneers in this field was a scientist named Otto Lowy, who conducted a series of elegant experiments that demonstrated the existence of acetylcholine.
Okay, I'm intrigued. Tell me more about these experiments.
So Lowy was studying frog hearts,
Okay.
and he had this hunch that the vagus nerve, which slows the heart rate, was releasing a chemical substance to kind of exert its effects.
Okay.
So he devised this clever experiment using two frog hearts.
Two frog hearts.
Two frog hearts. He isolated the hearts and placed them in separate chambers, each filled with a saline solution. The first heart was still connected to its vagus nerve. Lowy stimulated the vagus nerve,
Okay.
causing the first heart to slow down as expected.
Okay, so far so good.
Right.
What about the second heart?
Here's the brilliant part. He then transferred some of the saline solution from the first heart's chamber to the chamber containing the second heart.
And what happened?
The second heart, which had no vagus nerve connection, also slowed down.
Ooh.
This proved that a chemical substance released from the first heart's vagus nerve was responsible for the change in heart rate.
Wow.
Lowy named this substance Vogelstoff, which we now know as acetylcholine.
That's amazing. Such a simple yet elegant experiment
Right.
that led to a fundamental discovery.
It really is a classic example of scientific ingenuity. And this discovery opened the floodgates for research into acetylcholine and its role in the nervous system.
So once they knew acetylcholine existed, how did they figure out that there were specific receptors for it?
That came a few decades later with the development of new tools and techniques in pharmacology. Scientists started experimenting with different drugs and chemicals that could either mimic or block the effects of acetylcholine.
So they were essentially trying to find substances that could either amplify or silence the acetylcholine signal.
Exactly.
Yeah.
And through these experiments, they realized that there must be specific molecules on the surface of cells that were responsible for binding to acetylcholine and mediating its effects.
Ah, that's how they deduced the existence of acetylcholine receptors.
Yes.
But how did they differentiate between the nicotinic and muscarinic types?
Well, that's where things get even more interesting. They noticed that some of the drugs that affected acetylcholine signaling were influenced by nicotine, a compound found in tobacco plants.
So nicotine was somehow interacting with these receptors.
It was. And this observation led to the classification of acetylcholine receptors into two main types, nicotinic, which were sensitive to nicotine, and muscarinic, which were not.
So the naming was based on their sensitivity to this plant compound.
It was. And this distinction turned out to be incredibly important because it revealed that there were actually two distinct families of acetylcholine receptors.
Wow.
Each with its own unique properties and functions.
Okay, so we've got these two families, nicotinic and muscarinic, and we're focusing on the nicotinic ones in this deep dive.
Right.
But what about the specifics of nAChR subtypes? How did scientists figure out that there were different combinations of those alpha and beta subunits we talked about earlier?
That came about through a combination of techniques including molecular cloning and gene sequencing. Scientists were able to isolate the genes that encoded for nAChR subunits and study their structures in detail.
So they were literally looking at the blueprints for these receptors.
Exactly. And what they found was that there wasn't just one gene for each subunit. There were actually multiple genes encoding for different subtypes of alpha and beta subunits.
A whole family of genes for a whole family of receptors.
Precisely. And this genetic diversity explained why there were so many variations in nAChR structure and function.
It's like a mix and match system. Where different combinations of subunits create receptors with specialized properties.
That's a great way to put it. And this understanding of nAChR subtypes has been crucial for developing drugs that can target specific receptors, potentially leading to more precise and effective treatments.
It's amazing to think that we've gone from those early experiments with frog hearts to decoding the genetic blueprints for these complex receptors.
It really is a testament to the power of scientific inquiry. And this journey of discovery is far from over. There's still so much we don't know about nAChRs.
Really?
And each new insight opens up even more avenues for exploration.
So what are some of the big unanswered questions about nAChRs that scientists are still grappling with?
Well, one of the major challenges is understanding how the specific arrangement of subunits in an nAChR determines its properties. We know that different combinations of alpha and beta subunits can create receptors with varying sensitivities to acetylcholine, different ion permeability, and even distinct roles in various biological processes. But we're still piecing together the intricate details of this structure-function relationship.
So it's like having all the pieces of a puzzle, but not quite knowing how they all fit together to create the complete picture.
That's a great analogy. And another area of active research is understanding how nAChRs are regulated. We know that their activity can be influenced by a variety of factors, including other neurotransmitters, hormones, and even environmental cues. But we're still unraveling the complex interplay of signals that fine-tunes their function.
It's like trying to decipher the language of a complex orchestra, figuring out how each instrument contributes to the overall symphony of neural activity.
Exactly. And as we gain a deeper understanding of these regulatory mechanisms, it opens up possibilities for developing drugs that can modulate nAChRs activity in a more targeted and sophisticated way.
That's fascinating. Sounds like the future of nAChRs research is full of potential.
Yeah.
What are some of the most promising avenues for future research and therapeutic applications?
One area that holds a lot of promise is personalized medicine. As we learn more about the genetic variations in acRs and how they influence individual responses to drugs, we can potentially tailor treatments to each patient's unique profile.
So instead of a one-size-fits-all approach, we could have medications that are specifically designed for individuals based on their genetic makeup.
Exactly, and this could lead to more effective treatments with fewer side effects. Another exciting area is the development of new drugs that target specific nAChR subtypes.
Okay.
As we uncover the unique roles of these different subtypes in various diseases, we can design drugs that precisely modulate their activity, potentially offering more targeted and effective therapies.
It's incredible to think that these tiny receptors could hold the keys to treating such a wide range of conditions.
It really is. From Alzheimer's disease and Parkinson's disease to schizophrenia addiction and even chronic pain, nAChRs are emerging as promising targets for developing innovative therapies.
Well, I think it's safe to say that our deep dive into the world of nAChRs has been a journey of discovery, revealing their incredible importance in how our bodies function and the vast potential they hold for future medical breakthroughs.
I couldn't agree more. From their humble beginnings as a mysterious substance in frog hearts to their current status as cutting-edge targets for drug development, nAChRs continue to captivate our scientific curiosity and inspire new avenues of exploration.
And for you, our listener, we hope this deep dive has equipped you with a newfound appreciation for these tiny but mighty receptors and the vital role they play in the grand symphony of life. Keep exploring, stay curious, and remember, even the smallest discoveries can unlock the greatest mysteries.